Maths is a topic that you seem to either love or hate, a bit like Marmite. But for me, I love it, and I'm hoping to try and spread this passion to you. I'm going to help you improve your understanding by working through some examples and then giving you some questions to try yourself. Maths and biology makes up 10% of your marks, so it's really important that you don't neglect it.
So first of all, you are going to need some of these pieces of equipment. If you don't have any, just pause the video and go and grab it. So you need a scientific calculator, a nice sharp pencil, or a sharpener if you don't have a sharp pencil, some scrap paper, a rubber, and a transparent ruler, preferably one that's around 30 centimeters long. Okay, now that you've got all of your stuff, I'm going to give you an overview of the topics that we're going to be studying. So there are 5: arithmetic, data, algebra, graphs, and geometry. And I'm actually going to start with just some specific maths in biology. So this won't come up in your other subjects like chemistry and physics. The rest of it does overlap with chemistry and physics. So it's really good if you're also studying those because that'll come up across all 3. Okay, there are some symbols you're going to see. The EG means I'm going to take you through an example, so you can just watch and follow along. And the Q means that it's a question for you, so you can pause the video, give it a go, and then press play, and we'll go over it together. Number 1, maths in biology. Okay, so this is maths in biology. So we're going to start with some conversions. So you've got your transparent ruler in front of you. If we zoom in on 1 centimeter, we can see that it's divided into smaller units, and those units are called millimeters. So each little one is a millimeter. And we can see that there are 10 millimeters in 1 centimeter. So that's our first conversion. 1 centimeter is equal to 10 millimeters. Okay, so we're going to look at the next smallest unit, and that is called a micrometer, or a micrometer. There's 2 ways to pronounce it. And it has this funny symbol. This is called mu, which is just a Greek letter. So how many micrometers do you think are in 1 millimeter? It's a lot. There are 1,000 micrometers in 1 millimeter. Okay, but it gets smaller than that. We've also got something called a nanometer, or nm. And how many do you think are in 1 micrometer? Well, it's actually the same. There are 1,000 nanometers in 1 micrometer. Okay, so those are generally used for really, really tiny things like nanoparticles or things the size of a virus. So let's look at some common conversions. You're going to get microscopy questions, and often you'll be changing between image sizes and actual size of cells. So this one, from centimeters to millimeters, is really common. The first thing you have to do, if you go this way, is you just multiply it by 10. And if you want to go the reverse way, you're just going to divide by 10. So it works the same way. This is our second most common conversion that you'll have to do. Nanometers, they don't tend to come up as much, but they can do. So we'll do the conversion from 1 millimeter into 1,000 micrometers. This time you can see that you're just going to be multiplying by 1,000. So if you want to go back the way, you're just going to divide by 1,000. Okay, so let's have a go at working through these together. And then when we go into microscopy, we're going to put this into action with some questions. Okay, so if you're confident, you can pause the video and try them, and then we'll go through the answers. Ready? Okay, so we're going to go from millimeters back to centimeters. And remember, when we go back, we are going to be dividing. So we're going to divide by 10 when we go this way. So with your calculator in hand or using your head, you can see that if we divide by 10, the decimal point moves 1 to the left. So it's going to be 0.21 centimeters. We're also going to convert this number into micrometers. So we're going forwards, or we're getting smaller in our units. So we're going to be multiplying, and this conversion is by 1,000. Okay, so if we multiply it by 1,000, we get 2,100 micrometers. Okay, now we're starting with the micrometers and we're working this way. So if we're working this way, will we be multiplying or will we be dividing? Yeah, we'll be dividing. Okay, so if we go back this way, we are going to divide by 1,000. And then when we go back one more step, we're going to divide by 10. Okay, so this number is going to get really, really small. So it's going to be 0.078 because we've moved our decimal point 1, 2, 3 times. It's going to get even smaller again. It's going to be 0.0078 when we turn it into centimeters. Okay, and now the last one, we're starting with centimeters and we're going to work our way across getting into the smaller units. So this time, instead of dividing, we're going to be multiplying. So over here, we will see that that is 6 millimeters. And you can check this one on your ruler. You can see not a whole centimeter is going to be 1, 2, 3, 4, 5, 6 millimeters is less than 1 centimeter. So that makes sense. And then we're going to do it again. We're going to multiply it by 1,000. And so we get 6,000 micrometers. Okay, now we're going to look at microscopy. And you can see we've got an equation here. This is an equation that we need to memorize for the exam. And if you like using triangles, there's an easy way to learn this. Memorize. Just checking I spelled that right. Okay, so at the top, we're going to have I for image. That goes up here. Then at the bottom, we've got real object, which a lot of the times you'll see written as the actual size. So the size of the actual thing you're viewing. And then we've got magnification, which we can shorten to M. So you can see that the triangle says I am. So that's an easy way to try and remember it. And then the triangles work like this. This line across the middle means divide. And this line here means multiply. So if you, for example, wanted to find the size of the image, you just cover that up and then you work with the other two variables. So it would be actual size times magnification. Let's say you wanted to find the magnification, you would just cover that up and then you work with the other two variables. So it would be I, or the size of the image, divided by the actual size of the object, or the real size. And the same finally for the actual size. You cover it up and it becomes image size divided by magnification. Okay, so I'm going to do an example and then you're going to try this question. So a picture of a cheek cell measures 28 millimeters when magnified 300 times. Calculate the real size of the cheek cell in micrometers. Okay, so we've got two different units here. This one's in millimeters and we're going to have to give our answer in micrometers. So we're going to make sure that the units are the same. What we're going to do is turn these millimeters straight away into micrometers. So 28 millimeters, we need to turn it into micrometers. Do you remember the conversion? Yeah, it's just times by 1,000. So we're going to have 28,000 micrometers. Okay, so that is the size of the picture of a cheek cell. Now, if it's a picture, is it the image or is it the real object? Yeah, it's the image. So we've got the image is our 28,000. That's the eye, the image. And we've been told the magnification is 300. So we go over to our triangle. We want the A, so we're going to do I divided by M. And when we put our numbers in, it's 28,000 divided by 300. And you can do that on your calculator. And it gives you 93.33333 recurring. Okay, and that's your answer in micrometers. Okay, now you try, pause the video, and then press play when you've had a go at the question. Ready? So this mitochondrion is 10 micrometers long. When it is magnified, it measures 8 millimeters. How many times has it been magnified? So we're looking for the magnification. So it's going to be magnification is image divided by actual. So we can just write that down to remind ourselves right now what we're going to do. And we're going to be working with two numbers that again are different units. So we need to make them the same. It actually doesn't matter whether you turn them both into millimeters or you turn them both into micrometers, as long as they're the same. So let's turn our image size into micrometers. At the minute, it's 8 millimeters. So when we turn it into micrometers, we're just going to multiply by 1,000 again, and it will give us 8,000 micrometers. Okay, so now we can put our numbers in. Magnification is image over actual. So we're going to do 8,000 divided by 10. And that is simply going to give us a magnification of 800 times. You don't need to write anything after it, it's just 800. Okay, so here's another specific part of maths in biology. And this one is actually just for people studying higher tier. So if you're studying foundation, skip ahead to the next section. So this one is the light intensity and inverse square law. And this is about the relationship between distance and light intensity. As the distance increases, the light intensity decreases. That makes sense because as you get further away, the light intensity will get weaker. But this is not linear, which means if we plotted it on a graph, it wouldn't be a straight line. Instead, the light intensity changes in inverse proportion to the distance squared. We write it like this. Light intensity is proportional to 1 over the distance squared, which means it's inversely proportional. That might sound a bit confusing, so let's take a look at an example to see what this actually means. If distance is doubled, what happens to the light intensity? So when we say something is doubled, we mean it's twice as much. So we're going to put a 2 in place of the distance, and then it gets squared. So it's 1 over 2 squared, and 2 squared is just 4. So this means that the light intensity is as quarter as strong as it was before the distance was doubled. So it's got weaker. Now you try the next two questions. Ready? If distance is tripled, what happens to the light intensity? So tripled means it's 3 times as far. So we do 3 squared, which just means 1 over 9. So the light intensity is 1/9 as strong as it was before that. And finally, if distance is halved, what happens to the light intensity? Half is 0.5, and 0.5 squared is actually a quarter, or 0.25. Let's do a bit of math here. When you divide by a quarter, like this, 1 divided by a quarter, it's actually the same as flipping the second fraction and multiplying it. So 4 over 1. And 1 times 4 is just 4. So that means that the light intensity gets 4 times as strong. So you're bringing the distance of the light closer, so it's going to get stronger. So 4 times as strong. I hope that makes sense. Number 2, arithmetic. Now we're going to look at decimals. You could be asked to give your answer to 1 decimal point, 2 decimal points, 3 decimal points, etc. So this is our decimal point. And you always look at the number after the decimal point that you've been asked for. So when we're asked for 1 decimal point, we're actually going to look at the 7. And the 7 will determine whether or not the 4 stays as a 4 or whether it rounds up. And the rule is, if the number is 5 or greater, i.e. 5, 6, 7, 8, or 9, then you just round up. So in this case, we've got a 7, which is greater than 5. So it's going to make the 4 round up and become a 5. So to 1 decimal point, this number is going to be 5.5. Okay, so for 2 decimal points, remember you always look at the number after. So for 2 decimal points, we're going to look at the 9. And the 9 is definitely greater than 5, so it's going to make the 7 round up and give us an 8. So to 2 decimal points, it's going to be 5.48. 3 decimal points, let's pretend we have a 3 there. So we're going to look at 1, 2, 3, the number after. We look at this 3. 3 is less than 5, so we are just going to leave the 9 as it is. So that would just give us 5.479. Okay, pause the video and try these questions yourself. Ready to go through them? Okay, we're going to start by rounding to 1 decimal point. So we're going to look at the 2. The 2 is less than 5, so we're going to leave the 3 as it is, and it gives us 4.3. 2 decimal points, we look at the 5. This is 5, so we're going to round up, and that 2 becomes a 3. So it's 4.33. 3 decimal points, we're going to look at the 8, and the 8 is greater than 5, so we're going to round the 5 up to a 6. 4.326. Okay, the next one, we have got 7.9649. So to 1 decimal point, we'll look at the 6, and the 6 is greater than 5, so it's going to make the 9 round up. But when a 9 rounds up, it gives us 10. So the 10 is going to carry forward to the units, giving this 7 and 8 instead. But for our 1 decimal point, we still have to include it, and that's just going to be a 0. So make sure you don't just leave that off, you do need to include it. Okay, to 2 decimal points then, we look at the 4, that's less than 5, so it's not going to do anything to the 6, so it's just 7.96. And to 3 decimal points, we've got a 9, so that makes the 4 round up to a 5. 7.965. Next up is standard form, and this is used to represent really big or really small numbers. It's written as a decimal number, and that decimal number needs to be between 1 and 10, so we can't have anything less than 1, and we can't have anything bigger than 10. And it's then multiplied by 10 to the power of something. We'll use x to just show that it could be anything. So, for example, it could be 4.2 times 10 to the power of 4, or it could be 3.98 times 10 to the power of minus 6. Now, the sign in front of the power matters. When there's no sign, it just means there's an invisible positive. And a positive sign means that it is a big number. When you've got a negative sign, or that little minus, it means it's a really tiny number or a decimal number. So let's look at some examples and you'll understand that a bit better. So we need to turn this number into standard form. And the first thing we need to do is identify what our decimal number between 1 and 10 is. So you're going to place a decimal point somewhere that gives you a number between 1 and 10. So in this case, we're going to put it here between the 3 and the 6. So we get 3.6. After it wouldn't work because that would be 36. And before it would be 0.36, which is less than 1. Okay, so now we've got 3.6 times 10 to the power of something. And we work out what that something is just by counting the number of hops between this decimal and the real decimal, which would be here. So we just count 1, 2, 3, 4, 5. And that tells us it's 3.6 times 10 to the 5. If this is small enough, you'll be able to type it in on your calculator and see that it gives you that answer. Okay, so let's do the next one. We're going to put our point somewhere that makes it a number between 1 and 10. And we can see that it would fit in well here, 4.1. So let's count the hops. This time we're going backwards. 1, 2, 3. So it's going to be 4.1 times 10. 4.1 times 10 to the 3. But remember, we went backwards and it's a small number, a decimal number, so it needs to be a minus 3. Okay, and now the last one. Again, let's place our point. It needs to be in here, making 5.82. And now we're going to count what our power is. So we're going backwards, so it's going to be a minus. 1, 2, 3, 4. So minus 4. Okay, pause the video and try your questions and then press play. Ready? Okay, so for this one, it's 1, 2, 3, 4. So it's 7.8 times 10 to the 4. For this one, it's 1, 2, 3, 4, 5, 6. So 6.21 times 10 to the 6. And here, it's got to go here and be 1, 2, 3. 1.05 times 10 to the minus 3. Check you got that minus. So this time we've now got ratios, fractions, and percentages. These are all grouped together on the specification, so we'll go through each one in turn. So starting with ratios. These tell us a relationship between two things. For example, if the people to cake ratio was 3:1, that would mean that you had 3 people for every 1 cake. That's pretty good, eh? So this is a simplified ratio because one of the numbers is 1. All right, let's see how we can write and simplify ratios. So here's our example. 30 centimeters cubed of amylase solution is mixed with 6 centimeters cubed of glucose solution. Calculate the simplest ratio of the volume of amylase solution to the volume of glucose solution. And the wording in that question is important. Whatever they say first is what we're going to write first. So they say amylase first, which means we start with our amylase number, and that is 30. So it's going to be 32, and then our glucose number, which is 6, 30 to 6. Okay, but it wants a simplest ratio, and remember, when we say it's the simplest, that means one of the numbers is 1. So there's 2 ways you could do that. You could divide both numbers by 30. 30 divided by 30 is 1, and then we'll get an answer for what the 6 becomes. Or you could divide both numbers by 6, because 6 divided by 6 is 1. Now, it's best to do it in such a way that you end up with whole numbers. So in this case, it would be best to divide both sides by 6. So that's what we'll do, divide by 6. So 6 divided by 6 is 1, and 30 divided by 6 is 5. Okay, now it's time for your question. Pause the video and press play when you've tried it. Ready? Okay, 60 centimeters cubed of water is mixed with 15 centimeters cubed of acid. Calculate the simplest ratio of the volume of acid to water. So we need to give the acid number first, which is 15. So we're going to do 15. And then the water number, which is 60. 15 to 60. Okay, again, it wants the simplest ratio, so we need to get it down to 1. So we divide by the smaller number, which is 15. And when we divide both sides by 15, we're going to get 1 to 4. So that is your acid to water 1 to 4. Now we'll look at fractions. So fractions are when you've got a number over a number, another number. So here we've got A divided by B. A is our numerator and B is our denominator. So you're going to get questions in science that ask you to work out a fraction of something, normally a quantity of something. So the way that you're going to do it is you take your quantity, whatever it is you're starting with, you divide it by the denominator, and then you multiply it by the numerator. If you've got a calculator, you can do this all in one go, just put some brackets in. Okay, so let's look at an example. A student measures 36 milliliters of water, then adds 2/3 of it to the beaker of HCl. How much did they add? Okay, so our quantity is the 36. We've got 36 milliliters of water. And then we want 2/3 of it. So what we're going to do is we're going to divide by the denominator. 2/3 looks like that. So the denominator is 3. So we do 36 divided by 3. And that gives us 12. And then we want the 2 of it, and that's the numerator. So we're going to do 12 times 2, which just gives us 24. If you've got a good calculator, you can just do this all in one go and just do 36 multiplied by 2/3. And that'll give you the exact same answer. And don't forget to put your units on at the end, milliliters. Okay, pause and try the questions, press play to go through the answers. Ready? A student weighed 42 grams of sugar, then added 6/7 of it to 200 mils of hot water. What mass of sugar is in the water? So we need to focus on the sugar number, because that's what the question says, and it's 42. So we're going to do 42 times 6/7. So remember, you can do 42 divided by 7 and then multiply it by 6. or you can just go straight into your calculator for this one. And that will give you 36. Next one, a student uses 25 mils of amylase solution in an experiment, then repeats it with 3/5 more amylase. How much amylase solution was used the second time? Okay, so we start with our 25, that's our quantity, and we're going to multiply it by 3/5. So we can do 25 divided by 5, which is 5, and then times it by 3, which is 15. Or you can put it all in on your calculator and see that answer. Now, read the question carefully. This isn't our final answer because it says how much amylase solution was used the second time. That's the total amount. It doesn't say how much more was used. It just says how much was used. So we need to go back and add the 15 to the original 25 to get our final answer. And when we add those together, we get 40 milliliters. So well done if you got that and spotted that. Okay, and then we've got percentages. So this is working out the percentage from a quantity. So I'm just going to do some examples on the spot here. Let's say we want 11% of 35 milliliters of acid or something. So all you do is you take your quantity number and you divide it by 100 to get 1%. And then you just multiply it by how many percent you have, which is 11. So if you do that on your calculator, you get, let's see, 3.85 milliliters. If you're confident with decimals and percentages represented by decimals, you could also have done this as just simply 35 times 0.11. That's just turning 11% into its decimal form. And again, that gives you the exact same answer. So whichever way you're confident with. Okay, let's do another example. This time we want 0.25% of 78 centimeters cubed. So this time, again, we're going to do the same thing, 78 divided by 100. And then times by 0.25. And if you do that in your calculator, you're going to get 0.195 centimeters cubed. For your calculator, if you want to do it this way, you could also turn 0.25 into percentage. Just be careful. So it's less than 1%. So we're not even going to have a 1 here because that would be 1%. It's another 0 and then your 25. And that gives you your answer. Okay, try these two questions on your own. Read them carefully. There's a little bit of a trick here. Ready? Okay, first one. A potato chip was weighed and then placed in water for 3 hours. Its mass increased by 12%. Work out its new mass. Okay, and you needed to read the number here from the scale. So this is its starting mass. It's 24.34. Okay, increased by 12%. So we want another 12% of it. So we're just going to take this number and we're going to divide it by your 100 and then multiply it by 12, which will give us our 12. And that is 3.282. And then because it says we want the new mass, that means we need to add this on to the old mass. So we need to add these two numbers together, 3.282. And you can use your calculator for that. Let's see what we get. It's 30.622 grams. I will just mention the other way of doing this part of the equation. Instead of dividing by 100 and multiplying by 12 and then adding this on, you could actually just have gone straight to doing 27.34 times 1.12, basically 112%, and that will just give you this final number directly. Okay, another potato chip weighed 26.20 grams at the start and 28.32 grams at the end. Work out the percentage change in mass. Okay, so we're working backwards here. We need to get the percentage change. So let's start by working out what the change is in grams. Okay, so we're going to do 28.32 minus 26.20. And you could do that on your calculator or in your head. This is a nice one. And we get 2.12 grams. We want that as a percentage change. So we need to do it from our start value. So we're going to do 2.12 divided by 26.20 and then times it by 100. Let's do that on our calculator and see what we get. Divided by 26.20 times 100, and that gives us 8.09. Estimates are a quick way to see if your answer is correct. They're done without a calculator in your head, and it's to make sure that whenever you've typed something on your calculator, you haven't hit a button by mistake. So, you need to make sure that you use numbers that you're going to be able to calculate in your head. The suggestion is to round it to a number near to 10 or just a nearest whole number. So in this example, 0.75, you could round that to 1 and you're multiplying it by 9.2, which you could round to 10. So you know that your answer is going to be close to 1 times 10. It's going to be close to 10. Okay, the next one, we've got 122. You could round that to 120 or even 100 if you prefer to work with that. And 2.34 we could round to 2. So when we do 120 times 2, we get 240. Try the next two yourself. Ready to go through them? Okay, a student uses a potometer to measure the rate of transpiration. The bubble moves 0.46 millimeters per minute. Estimate how far it moves in 19 minutes. Well, starting with that, we could round that instead to 20. And 0.46, we could probably round that to a half, or 0.5. So it moves half a millimeter every minute for 20 minutes. So we multiply them together. And there's a couple of ways to do this. You can think of 0.5 as a half. So half multiplied by 20. And if you're good with multiplying fractions, you just multiply the top numbers. So it becomes 20 over 2, which is 10. Or you could think of multiplying it by 2 and then by 10. So when you do 0.5 times 2, that gives you 1, and then you just times it by 10, which again gives you 10. Either way, hopefully you got the answer of 10. And the second one, in a study of 103 people, 11% were found to be at risk of type 2 diabetes. Estimate how many people are at risk. So 100 people is a good number to round to. And then we want 11%, 11% of 100 is just going to be 11. Or you could have rounded that to 10%, and 10% of 100 is just 10. So it doesn't really matter, whichever answer you got for that. So if you finish your exam with some extra time, it can be a good idea to recalculate any calculation questions you have and to carry out a rough estimate in your head. Number 3, data. Significant figures are something you could be asked to give your answer to. For example, 3 significant figures, 2 significant figures, or 1 significant figure. Here's the rule. You start counting from the first non-zero number, i.e. 1 to 9. Any zero that comes after that point counts as a significant figure. And remember to round your last number if it's greater than or equal to 5. Okay, let's take a look at an example. Here we figured out an answer to a question, and it is 67,342. And you've been asked to give your answer to 3 significant figures. So we start counting from the first non-zero number, which is the 6, and we're going to give 3 significant figures, so the first 3 numbers. So it's 6, 7, 3, and you fill up the remaining placeholders with zeros, 0, 0, so that the number retains its correct size. Okay, so for 2 significant figures, we're going to do the first 2 numbers, 6, 7. A reminder that you check the number after it to make sure it doesn't round up. It does if it's 5 or greater than 5. So it's a 3, so it doesn't round up. So it's 6, 7, and then we fill up the remaining numbers with zeros. Okay, and finally, one significant figure is just the first value. This time, the 7 is going to make the 6 round up because it's 5 or greater than 5. So we're actually going to end up with a 7, and then we fill up all of the rest of the numbers with zeros. So it's 70,000. Okay, we'll do one more example. This time it's going to have some decimal points. So give your answer to 3 significant figures. So remember, we start counting from the first non-zero number. So we don't include this, we don't include this, and here is where we start. We still need to put these numbers here, but they're not significant. So we write them down 0.0, and then we count 1, 2, 3. So we have 1, 0. And then the 6 is going to round up. Because the 8 follows it, and 8 is greater than 5, the 6 becomes a 7. So I've done this one on purpose because it's got a 0 in the middle. Remember, any 0 that comes after a number is significant. So this one is significant. Okay, now 2 significant figures. So again, we don't include the first 2. They still need to be written down, but that's not where we start counting from. So it's going to be 1, 0. And then the 6 comes after it, and 6 is again greater than 5. So the 0 is going to round up to a 1. So it's going to be 1, 1. And finally, one significant figure. We're going to start with our 0.0, and then we have got the 1. 0 means that it doesn't round up. So it looks like that. Okay, so now pause the video and try some of these questions yourself, and then press play to go through them. Ready? Okay, to 3 significant figures, this number is going to be 305 because the 7 has made the 4 round up. To 2 significant figures, it's going to be 300. And to 1 significant figure, it's going to be 3, and then it's just followed by 2 zeros again. It can happen that it looks the same. Okay, and the next one, 3 significant figures. 1, 2, 3. The 4 means it doesn't round up, so it's 8, 9, 6. To 2 significant figures, we're going to go 8, 9, but then the 6 is going to make the 9 round up. And if you recall from the decimals, when a 9 rounds up, it makes a 10, which makes this number now become a 9, 0.9. And then because it's 2 significant figures, we're going to have to give the 1 after it as well, which has just become a 0. Okay, and finally, 1 significant figure. We're just looking at this 8. And then again, the 9 makes it round up, and it becomes a 9. So 0.9. Now we're going to look at mean, median, and mode, and we'll just jump right in with an example. So students collected data on the heights of 5 shoots grown, that should say in, acidic soil. And here are their results rounded to the nearest centimetre. First of all, let's find the mean. So all we're going to do is add up all of the numbers, and then we're going to divide by how many numbers we have. We've got 1, 2, 3, 4, 5 numbers, so we'll divide by 5. Use your calculator for this, and you should get 3.2. Now we'll work out the median, which just means the middle value. So we need to take our 5 results and put them in order from smallest to largest. So it's 2, 2, 3, 4, 5. And then we just count our way into the middle. There's 2 on this side and 2 on this side. So our median is 3. The mode then is the most common value. We can see we've got 2 2's, so the mode is 2. And the range is just the biggest number take away the smallest number. So 5 take away 2 gives you 3. Okay. Now let's try the question yourself. Pause and give it a go. Ready? Scientists investigated the number of trees with the disease in 7 different woodlands. Here are their results. Find the mean and give your answer to 2 significant figures. So we're going to add up all of our numbers. Just a reminder, even if you do it in your head, always check it on a calculator because you never know, with that exam pressure, you can make a mistake. So when we add them up, we get 50 divided by 7 gives us a long number. We want it to 2 significant figures. So we want 7.1, and then the 4 means that the 1 doesn't round up. So we just leave it as 7.1. Okay, the median means we need to put our numbers from smallest to largest and then find the middle value. So 3, 4, 6, 8, 8, 9, 12. 1, 2, 3, 4, 5, 6, 7. It's always good to check that you got all of your numbers. And then the middle number will be the fourth one, 1, 2, 3, 4. So our median is 8. The mode then is the most common value, and that's actually going to be 8 again. We've got 2 of those. And the range is the largest number, which is 12. Take away the smallest, which is 3, which gives us a range of 9. So here are the rules for drawing frequency tables. First of all, make the intervals or classes the same size or width. Secondly, make sure that these intervals don't overlap, i.e. the numbers are different. And finally, aim for no more than 6 classes. Okay, so let's look at an example. We're going to complete the frequency table below for these results. So we need to first of all make our intervals or classes. Okay, so the biggest number is 6, the smallest number is 0, and we can see that we've got a nice range within that. Okay, so we'll start by splitting it into 0 to 1. We need to make sure they don't overlap. So then we'll go 2 to 3, 4 to 5, and 6 to 7. So those fit in nicely. Okay, now we'll go over to our frequencies, and we're just going to put the numbers in here. So 0 to 1, we've got 1, 2, 3, 4 that fit in that category. 2 to 3, 1, 2, 3, 4 that fit in that category as well. 4 to 5, we've got 1, and 6 to 7, we've got 1 as well. A good idea is to just tot these up quickly. We can see we've got 10, and then check that that's how many you should have. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Perfect. Okay, pause and try and do this question yourself. Ready? Okay, so first of all, we're going to complete the missing interval. So we just choose the next number up, which is 15, and the one below 20 is 19. We can check the interval matches the rest. 15, 16, 17, 18, 19 is an interval of 5. We can see the rest of them follow that as well. Okay, so now the frequencies, 5 to 9 would be 8, 7, and this 8, so 3 in total. 10 to 14, 1, 2. 15 to 19 is just 1. And then 20 to 24, 1, 2, 3. And again, we'll tot them up. 5, 6, 7, 8, 9. We've got 1, 2, 3, 4, 5, 6, 7, 8, 9. Perfect. You need to know some sampling principles. You'll already have covered transects and quadrats in the ecology topic, but in the maths part of your spec, this mark release recapture method does come up. So I'm going to teach you what it is and then how to use this equation to work out estimated population size. So the first step is that you go out and you collect a sample of something like these little snails. That's the capturing. Then you mark them, and that's just using something like white paint. So you can see this snail has got some paint on its shell now. Then you're going to release the snails that you captured, and you're going to leave them for a certain length of time, maybe a few hours or a few days or even a few weeks. Then you return to your sample site, and then you collect them all over again. So you collect a certain sample size, and then you see how many of them that you find have still got the mark. So let's do an example. Let's say the first time you go out to collect a sample, you find 20 snails. And then you mark them all. Then you leave them for, let's say, 2 days, and then you go out and you collect another sample size. This time you find 30. But of that 30, only 3 are marked with the white paint. So now we can work out what our population size might be. So we do 20 times 30, which gives us 600. And then you divide it by the number of recaptured marked individuals, which was 3. And you find out that we have an estimated population size of 200. Okay, so now it's your turn. Pause the video and try this question and press play to go through it. Ready? A class collected and marked garden snails in their school field. They found 30. They returned a week later and found 40, of which 4 were marked. Estimate the population size of the garden snails. So we start with the first sample size, which was 30, and we multiply it by the second sample size, which was 40, and then we divide it by the number of recaptured marked individuals, which was 4. So 30 times 40 is 1,200, divided by 4 gives us an estimated population size of 300 snails. You need to work out simple probability in biology, and it's most likely this is going to come up within the inheritance topic. So that's what we're going to look at for our examples. If you haven't already revised this, check out my inheritance video to work out how to do Punnett squares first. Okay, so here's our example. 2 heterozygous parents have a child. What is the probability of it having blue eyes? We can see that brown eyes is dominant because it's got the capital letter, and that blue eyes is recessive because it's got the lowercase letter. So they're heterozygous, which means they have one of each allele. So we're going to split up each parent's alleles to work out what they could pass on to their child or offspring. So we'll start with dad, big B, little b. You don't have to write the parents' names there because they're the same, and mum is also big B, little b. Now we just do our genetic cross, big B, big B, big B, little b, big B, little b, and 2 little b's. And now we can work out the probability of it having blue eyes. So the only child that could have blue eyes is this combination of alleles, and you can see that that's just 1 out of 4 possible combinations. So the probability of blue eyes is 1 in 4, or 1/4. You can write it either way. Okay, pause the video and try the question yourself. Ready? Okay, so a homozygous recessive plant and a heterozygous plant are mated. What is the probability of its offspring having purple flowers? So we can see that the big P for purple is our dominant allele, and the little p means that white flowers are recessive. Okay, so a homozygous recessive plant. Homozygous means 2 of the same alleles. So if it's homozygous recessive, that's 2 little p's. Little p, I'll draw my grid first, that would help. Little p for one plant. And then a heterozygous plant means they've got 1 of each allele. Big P, little p. Try and make your dominant ones look as big and capital as you can. So now we do our cross. We've got big P, little p, big P, little p, 2 little p's here, and 2 little p's here. What is the probability of its offspring having purple flowers? That is these 2 because they have that dominant purple allele. So it starts off with being 2 out of 4, and you can then simplify that to being a half, or 1 in every 2 offspring. Data can be represented with bar charts and histograms. We're going to look at the difference between them. First of all, this one is a bar chart. You'll notice that the gaps between the bars are equal and that the bars themselves are equal in width. This is because the data here is discrete. That means it has an exact value. For example, the number of apples, or it could be the data is actually written in words. For example, blood groups. And when it's written in words, we call it qualitative data. Over here, we've got a histogram. You'll notice that instead of having frequency up the side, it's got frequency density. The frequency is worked out by the area of each bar. And you'll notice that the bars can have different widths, which is the class width. We'll look at how we work this out in a moment. But finally, we'll mention that histograms are for continuous data. This is when the data can take any value, for example, mass or weight. So let's take a look at this example. I'll do 2, and then you're going to try these 2. So we've been given the frequency, and we know what the class is, but we don't know the width yet. So we'll work that out by doing the upper part of the class minus the lower part of the class. So 100 take away 50 gives us 50. Now to work out the frequency density, we're just going to take the frequency and divide it by the class width. So we're going to do 60 divided by 50, and that will give us 1.2. So now let's look at how we draw this on our graph. We've got the class from 50 to 100, so our bar is going to be across here, and we know now that it needs to go up to 1.2, which is approximately there. Okay, let's go back to our next one. Let's work out the class width from 100 to 150. That's a width of 50. And we're going to work out the frequency density by doing 75 divided by 50, which gives us 1.5. And again, we'll draw that on here. The class width is 100 to 150. And this time we want 1.5, which is roughly, let's say there. Okay, pause the video and now you try these two. Ready? Okay, so the class width this time is only 25. And when we do 65 divided by 25, we get a bigger number, we get 2.6. So let's draw that on there. And remember this time, we really want to go halfway. We want to go to 175 and then up to 2.6. Something like that. And our final group then is going to be a larger one. It's going to be 75 for the class width. And 180 divided by 75 gives us 2.4. So this is a big group and it needs to go all the way up to 2.4. Something like that. Okay, so that is what your histogram might look like. You can also represent data using scatter diagrams, and these are more commonly used in science. So we're going to look at 3 different scatter diagrams and what they can tell us about the relationship between the 2 variables. The first thing that you always do is plot a line of best fit. So you want to use a ruler for this. I always suggest your transparent ruler. And the rule is that you want to try and get as many points above the line as below the line, and you also want to get your line going through as many of the points as possible. So use a sharp pencil and make sure that you line it up before you draw it. So let's see, that goes through 3 points and that leaves 3 above it and 3 below it. So I think that looks pretty good. Because this is going up, we say that this line or this relationship shows a positive correlation. And you could describe that if you knew the names of the 2 variables, as saying as variable x goes up, variable y also increases. Okay, let's draw our line of best fit for this one. Why not pause the video and hold your ruler over the graph that you see, and let's see if it lines up with where I put my line. Ready? Okay, so there are lots of points this can go through. That leaves 2 below and roughly 2 above. And we would describe this relationship as a negative correlation because as variable x increases, variable y actually decreases. Okay, in this scatter graph, we've got points pretty much all over the place. It's going to be really hard to try and find a line of best fit. So in this case, we say that there is no correlation between our 2 variables. Just a reminder that just because there is correlation, it doesn't mean that there's causation. For example, in the summer, people buy ice creams and they also buy swimwear. That doesn't mean that ice creams cause people to buy swimwear. Orders of magnitude are used to make approximate comparisons between numbers or objects. Here's how they work. The first step is to divide the bigger number by the smaller number. If the answer is less than 10, then they're the same order of magnitude, i.e. they're around the same size. But if the answer is around 10 itself, then it's 10 to the 1 or 1 order of magnitude bigger. And if the answer is around 100, then it's 10 squared or 2 orders of magnitude bigger, and so on. So for this example, we are asked to calculate the order of magnitude comparison between this phone and the ant. So here's our phone and here's our ant, and we divide the bigger number by the smaller number. So we're going to do 14 divided by 1.2, and if we work that out, we get 11.6 recurring. That number is pretty close to 10, so we can say this is 1 order of magnitude bigger. I'm going to shorten that to order of magnitude. Okay, and then we're going to compare it to the ladybird. So when we do a comparison, remember that we have to make sure the units are the same. This ladybird is in millimeters, so we need to turn them into the same units. It doesn't matter if you turn them both into centimeters or both into millimeters, as long as they're the same. Let's turn 1.5 millimeters into centimeters. All we're going to do is divide by 10. So we're going to get 0.15 centimeters. It's less than a centimeter, so that number makes sense. Okay, so we're going to do 14, which is our biggest number, divided by the 0.15. And this time we get 93. And that number is pretty close to 100. So this time it is 2 orders of magnitude bigger. All right, pause and see if you can do these two questions. Ready? This time we're calculating the order of magnitude comparison between this bacterial cell and the virus. So you need to spot which one's the bacterial cell first. It's this one, and it's 1 micrometers long, and the virus is 100 nanometers long. So immediately we've got some different units. As we can see that the next one, the DNA, it's also in nanometers. It's going to be easier if we turn the micrometers into nanometers. So we're going to multiply by 1000 as our number is getting to a smaller unit, and that gives us 1000 nanometers. Okay, so now we're going to divide the bigger number by the smaller number. So we're going to divide the 1000 by the 100, and that will give us 10. And so our answer is going to be that it's 1 order of magnitude bigger. Okay, B, the piece of DNA. The piece of DNA is 10 nanometers, so we're going to do 1000 divided by 10, and this time it gives us 100, which just like before is going to give us 2 orders of magnitude bigger. Number 4, algebra. There are some algebraic symbols that you need to know. We'll go through each one and then we'll look at changing the subject. The first one here means that it is equal to something. The next one means that it's approximately equal to. This one means that it is less than. The double arrow means that it is greatly less than or a lot less than. When the arrow is flipped, it means more than. When we've got a double flipped arrow, it means it's greatly more than, so it's a lot bigger. And this one means it is proportional. We saw this when we looked at the inverse square rule, which is higher tier. Okay, so you just need to know what all of those mean. And now we'll look at changing the subject. So this is likely to come up within your microscopy equation, which again we looked at the start. So why don't you try this question and see if you can do it. Ready to go through it? Okay, so a cell has a diameter of 30 micrometers. It is magnified 200 times. Work out the size of the image and give your answer in millimeters. So let's isolate what we're working out. Size of the image, which is this one. Now, it's not the subject. The subject is whatever is on its own, followed normally by an equal sign. We want this to be the subject. So, if you remember your triangle, we can do it this way. I am is how we remember it. And the image is at the top, which is the I. So what we're going to do then is take the A and multiply it by the M. A, remember, we just meant actual, which is the same as the real object. Okay, so to work out the size of the image, we're going to do the magnification times by the actual. So we're just going to do 30 times 200. And that will just give us 6,000. Now, it then says that we want our answer in millimeters, because at the minute, this was in micrometers from there. So, to turn micrometers into millimeters, the rule is that we divide by 1,000, because we're moving into a bigger unit. So when we divide by 1,000, we just get 6 millimeters. Number 5, graphs. You could be asked to plot data on a graph drawing your own axes. First of all, always use a sharp pencil and a transparent ruler. And make sure your lines and points are clearly visible, as most exam papers are now scanned for marking. So use a dark pencil to be on the safe side. Here are the rules for drawing graphs. The independent variable should always go on the x-axis. This is the thing that changes, while the dependent variable should always go on the y-axis. This is what you record. You should also make sure that you stretch out your scale to use up as much of the paper as possible, and you should give labels with their units. Finally, you should draw a line of best fit. Remember, it doesn't have to go through the origin of 0, 0. It should go through as many points as possible and have as many points above the line as below the line. And if there's an obvious curve to the points, then draw a curved line of best fit following the same rules. Okay, so we're going to do an example of a graph. If you've got your graph paper to hand, pause and see if you can draw your graph, and then we'll press play to compare them. Okay, so remember, independent variable goes on the x-axis, that's what we're changing. So this one is going to be on the x-axis, and what we record, the dependent variable, goes on the y-axis. So let's pick our scale. It needs to go up to 1, and it starts at 0.2. And the first thing I always suggest is drawing your lines on where you can see the more bold lines. So I can draw my little markers here. And then I just tend to say them out loud. You can start writing them, but it saves rubbing out if you don't get your scale right the first time. So let's say, for example, we had 0 here, 0.1, 0.2, 0.3, 0.4, 0.5. That would mean we'd run out of space. So let's go up in a higher value or higher interval. 0.2, 0.4, 0.6, 0.8, and 1.0. That fits perfectly. Okay, we'll do the same up our other axes, drawing our little units, our little lines on the side, and then we can see that our lowest value is 1.2 and our highest value is 2.3. So let's say we start at 1, 1.2, 1.4, 1.6, 1.8, 2. That's not big enough. We need to go up in bigger values. So let's say 1.5, 2.0, 2.5, 3, 3.5. That goes up a little bit more maybe than we need, but if you start playing with things like 1.4, 1.8, it can get a little bit hard to work out the intervals. So we'll keep it simple. This uses up a large part of the graph. Okay, so let's go to 0.2 and plot the number of 2.3. You do need to count precisely your little points. So if it's going up from 2 to 2.5 and there are 10 little squares, that means we need 2 squares to be each 1 or 0.1, I suppose we should say. So we want 2.3, so we can count 4 little squares down. 1, 2, 3, 4, and we plot an x. The middle of the x should be where the point is. So do be careful about where your 2 lines cross. That's where it should go. 0.4 is 2.1. So we go to 2 and then 2 little squares up gives us 2.1. 0.6 is 1.7. So we can count 4 little squares up. 0.8 is empty. And 1.0 gives us 1.2. So we go to 1. 1.2 is 4 little squares up, so it's about there. Okay, and then we swap to our transparent ruler, and we're going to draw a line of best fit. This one looks quite nice. That looks pretty good to me. Okay, and let's make that line just a little bit thicker so you can see it. There we go. Okay, now you could get a question that then asks you to work out the value for 0.8. So pause and see what you would work out for that. Okay, so all we do is we go to our 0.8 and we read up and then we read across and we would get 1.5. Now there's one last thing we need to do to finish off this graph. Can you spot what it is? Yeah, we need to label our axes. So along the bottom, we've got the concentration of weed killer and we must give the units. It's M for moles. And along the side, we've got the growth of the plant. And if we were asked to describe this relationship, we could describe it as negative correlation, because as the concentration increases, the growth of the plant is decreasing. The equation of a straight line graph is always described by y equals mx plus c. M is the gradient, and we're going to look at how you work that out in a minute. And c is the y-intercept, which means where your line of best fit crosses the y-axis. So we're going to look at how you find that. And we're also going to mention that if your line of best fit goes through the y-axis at the point 0, 0, then that means it is directly proportional, because as one of your variables increases, the other one increases at the same rate. So that's a special relationship you need to know about. And remember, proportional had this little symbol like that. Okay, so let's look at our graph from the previous example, and you could be asked to work out the y-intercept. So we just go to our line of best fit, which was here, and we work out where does that cross the y-axis. We can see it's here, and if we count, that's 1, 2, 3, 4 little squares up, and each 2 squares was 0.1. So that means this is 0.2 added on to 2.5 gives us 2.7. So 2.7 is our y-intercept here. Okay, now try this question. Pause the video and give it a go. Ready? Okay, so can the relationship between height and mass be described as directly proportional? When we look at the line of best fit, it doesn't go through the origin point of 0, 0. So no, it cannot be described as directly proportional. They're not increasing at the same rate. And B, what is the y-intercept? So we're going to follow our line of best fit back to the y-axis, which is this one. And we can see that it is 65. And don't forget, you need to give your units, so 65 kilograms. Now we're going to look at how you can work out the gradient of a graph, which is the m part of the y equals mx plus c equation. The gradient is worked out by dividing the change in y by the change in x. So you just need to make a triangle out of your graph and then divide the y value by the x value. Here's an example for the lactic acid produced in a person's muscles during exercise before they take part in a training program, and we're going to compare it to after they take part in a training program. So it's going to be useful to see how much the lactic acid increases by working out the gradient and comparing those two numbers. So we're going to make our triangle over here going across and down. I always suggest you just use the entire line of best fit, because one of your numbers will lie alongside the actual values, making it easier to work out what that number is. Okay, so we need to first of all work out our scale. The difference between 4 and 2 is 2, and there are 10 squares. So we're just going to do 2 divided by 10, which gives us 0.2. So each little square is worth 0.2. I think this is about just one little square above the 2. So that number is 2.1. And now we'll work out what this top number is. It is 1, 2, 3 little squares below the 10. And each square is 0.2. So if it's 3 squares, it's 3 times 0.2, which is 0.6 below the 10, which actually gives us 9.4. So now we want to work out what this whole y side is. So our change in y is 9.4 take away 2.1, which gives us 7.3. Now we'll do the same thing for this x value. Over here we can see it's 0, and by the end it's 5, so that's an easy one. 5 take away 0 is just 5. So we've got both of our numbers, and now remember we're going to do y divided by x. So we do 7.3 and we divide it by 5, and that gives us 1.46. Okay, pause the video and see if you can work out the gradient for this line. Ready? So we just make it into a triangle. If you're anything like me, you'll just have used the entire line. And we'll work out our change in y first. So the end value is 6 and the start value was halfway between 0 and 2, which is just 1. So the change in y is 6 take away 1, it's 5. The change in x is from 0 to 5, so again it's just 5. This is an easier one. We do y divided by x, which is 5 divided by 5, and that gives us 1. We can see that before the training there was a much steeper increase in the amount of lactic acid produced, whereas after training it was a lot more of a gentle increase. It was a flatter gradient. And lastly, number 6, geometry. You could be asked to work out the surface area of different shapes. We'll start with the example of a rectangle. It's really easy, you just do length times the width. So we've got 15 millimeters and 3 centimeters. If we just multiply those numbers together, that's not going to work because we've got 2 different units. We need to make them the same. Again, it doesn't matter if you turn them both into millimeters or both into centimeters, as long as they're the same. Let's turn this 15 millimeters into centimeters. All we're going to do is divide by 10, so you get 1.5 centimeters. Now we can just multiply our numbers together to get our area, and it gives us 4.5 centimeters squared. Remember, area always gets a little squared. Okay, now we've got a triangle. A triangle is just half the base times the height. Okay, so we can just do 7 times 7 and then multiply it by half, which is the same thing as doing 7 times 7 and then dividing it by 2. So 7 times 7 is 49. And when we divide that by 2, we get 24.5 centimeters squared. Now, using what we've done for these two, see if you can pause the video and work out the surface area of this shape. Ready to go through it? Okay, so you need to divide it into a rectangle and a triangle, and then we can use these two equations that we've used before. We'll also need to do some unit conversions. So starting off with the rectangle, the length times the width. We've got 2 different units. Let's turn the millimeters into centimeters because everything else is in centimeters. So 25 millimeters, if we just divide by 10, gives us 2.5 centimeters. Then we're just going to times our 4 with our 2.5 to give us the surface area of the rectangle, and that's 10. Now we need to do our triangle. However, we don't quite have the values we need yet, so we're going to have to do some working out. This whole length is 6 centimeters, and this portion of it is 2.5 centimeters, which means that this remaining bit must be 3.5. We just get that by doing 6 take away 2.5 gives us 3.5 centimeters. It can make a bit more sense if you write it beside the triangle itself, 3.5 centimeters. Okay, we also need to work out this dotted line. The whole length here is 4 centimeters, and this part of it is 2 centimeters, which means that just this bit must also be 2 centimeters, because they need to add up together to give you 4. Okay, so now we've got those 2 values. We are just going to do a half times 2 times 3.5, and that gives us 3.5. The last step then is just to add them both together. 10 plus 3.5 gives us 13.5 centimeters squared. You could also be asked to work out the surface area of a cube or a cuboid. All you need to do is work out the surface area of each face and then add it together. There are always 6 faces. Another tip for you is to bring a little cuboid rubber with you into the exam, and then you can mark off which sides you've already added up. It can just help you visualize it a little bit better. Okay, so let's start with the end faces. And there are going to be 2 of those. You can see those in this diagram here, one here and one here. And we can see the dimensions. We've got 3 and 2.2. So we're just going to multiply 3 and 2.2 together, and that gives us 6.6. Now we're going to do the top and bottom faces, which there are 2, one here and one here. And we can see the dimension of the long side is 6, and this short side is 2.2. So we're going to do 6 times 2.2. And that gives us 13.2. And finally, we're going to do our two other sides. This one and the one that's on the back over here. So this one and this one. So again, the long side is the 6 centimeters, but this top side here is 3 centimeters. So it's going to be 6 times 3, and that gives us 18. Now remember, there are 2 of each of these, so we have to multiply each number by 2, and then we can add them together. So we've got 13.2 plus 26.4 plus 36. And we're just going to add all 3 of these numbers together, and that gives us 75.6. And our units are in centimeters, so it's centimeters squared, because it's still surface area. Okay, pause the video and see if you can work out the surface area of this cuboid. Ready to go through it? Okay, the first thing is that we've got some disparity with our units. We've got a mix of millimeters and centimeters. Because we've got 2 in millimeters, we'll turn the centimeters into millimeters to join them. So 3 centimeters is 30 millimeters. You can just check that on your ruler. Okay, so let's start by working out the front and back faces. We're going to do 30 times 45 gives us 1,350. Then we'll do the top and the bottom. So this side is 27 and this side is our 30. So 27 times 30 gives us 810. And then our last 2 sides, this side and this hiding side over here. The long side is our 45 and this side here is our 27. So 45 times by 27 is 1215. Then remember, we need to double all of the sides to give us our 6 faces. So we have 2,700, 1,620, and 2,430. And we finish up by adding them all together, which gives us 6,750 millimeters squared. The last thing you could be asked to work out in geometry is the volume of a cuboid. All you're going to do is the length times the width times the height. Nice and easy. But as before, make sure that you check your units. Here we've got a mix of centimeters and millimeters, so we'll change our 2.2 centimeters into 22 millimeters. Now we can just multiply our 3 numbers together. 22 times 7 times 5 gives 770 millimeters cubed, because it's now in volume. Okay, pause and try the next question yourself. Ready? This time we've got some meters just to mix things up, but this one's in centimeters. So we need to turn centimeters into meters. There are 100 centimeters in a meter, so we need to divide by 100 to get our units in meters, which is just going to be 4.9 meters. And then we can multiply our 3 numbers together. 7.2 times 5.6 times 4.9. And on our calculator, that gives us 197.568, a long number. And that is meters cubed.
Thanks for watching, guys. I hope you got a lot out of that maths and biology video. If you enjoyed it, please remember to subscribe and check out my other videos.
You can also support me by buying me a virtual coffee on ko-fi.com. The link's below. And thank you again so much for watching my videos.